So let's start with transcription. So the process of copying the genetic information from one strand of the DNA into RNA is termed as transcription. Here also the principle of complementarity governs the process of transcription, except the adenosine complements now forms base pair with uracil instead of thymine. So, however, unlike in the process of replication, which once set in, the total DNA of an organism gets duplicated, in transcription, only a segment of DNA and only one of the strands is copied into RNA. So, this necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. So why both the strands are not copied during transcription has the simple answer. First, if both the strands act as a template, they would code for RNA molecule with different sequences. Remember, complementarity does not mean identical, and in turn if they code for proteins, the sequence of amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Secondly, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would be a double-stranded RNA. So this would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. Now, transcription unit. A transcription unit in DNA is defined primarily by three regions in the DNA: a promoter, a structural gene, and the terminator. So there is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA-dependent DNA polymerase can catalyze the polymerization in only one direction, that is 5' to 3', the strand that has the polarity 3' to 5' acts as a template and is also referred to as a template strand. The other strand, which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil, is displaced during transcription. Strangely, this strand, which does not code for anything, is referred to as coding strand. All the reference point while defining a transcription unit is made with coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. So template strand, this is the template strand 3' to 5', and coding strand is from 5' to 3'. Can you now write the sequence of RNA transcribed from the above DNA? So template का उल्टा होगा, simple, just template को copy करना है, बस T की जगह पे U रहना है. No, you don't have to copy the template strand. इसका कॉम्प्लीमेंट्री लिखना है, लेकिन टी की जगह पे यू लिखना है, ठीक है? तो इसका क्या मतलब हुआ? इसका क्या मतलब हुआ कि ये वाला चीज़ जो है ये कोडिंग है एंड ये सेम रहेगा, ठीक है? ये सेम RNA रहेगा, जस्ट दैट टी की जगह पे यू रहेगा, ठीक है? So can you write the sequence of RNA? Okay, so you can write it. इसका कॉम्प्लीमेंट्री रहेगा RNA, लेकिन जहाँ पर T आएगा वहाँ पे U रहेगा, और ये भी तो इसका कॉम्प्लीमेंट्री है ना, तो ये सेम रहेगा, बस T की जगह पर U रहेगा. ओके, नाउ सिंस वी नो एवरी रेफरेंस हम लोग लेते हैं किसको है, कोडिंग स्ट्रैंड को. सो कोडिंग स्ट्रैंड इज फ्रॉम 5' to 3'. तो कोडिंग स्ट्रैंड का स्टार्टिंग मीन्स 5' एंड पे प्रोमोटर रहेगा एंड 3' एंड पे टर्मिनेटर रहेगा एंड बीच में जो रहेगा वो स्ट्रक्चरल जीन कहलाएगा. सो promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5' end upstream of the structural gene. Means this reference किस respect में ले रहे हैं? Coding strand के respect में. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strands. By switching its position with terminator, the definition of coding and template strands would get reversed. मतलब कि promoter अगर यहाँ पर हो जाएगा तो promoter कहाँ पर होता है? 5' end of the coding strand. तो ये coding strand हो जाएगा. फिर terminator अगर यहाँ आ गया तो coding strand ये हो जाएगा. ठीक है, ये दोनों एक्सचेंज हो जाएगा तो कोडिंग स्टैंड हो जाएगी और ये टेम्पलेट स्टैंड हो जाएगा. ठीक है, द टर्मिनेटर इज लोकेटेड टुवर्ड्स द 3' end means downstream of the coding strand, and it usually defines the end of process of transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. Now, the transcription unit and the gene. A gene is defined as the functional unit of inheritance. Though there is no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence. So the DNA sequence coding for tRNA or rRNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. So in eukaryotes, the monocistronic structural gene have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or expressed sequences are referred to as exons. So exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. So inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. Now, the types of RNA and the process of transcription. So, in bacteria, there are three major types of RNAs. One is mRNA, means messenger RNA. One is tRNA, means transfer RNA, and rRNA means ribosomal RNA. So all these RNAs are needed to synthesize a protein in a cell, mTR. The mRNA provides the template. tRNA brings amino acids and reads the genetic code. And rRNAs play structural and catalytic role during translation. There is a single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. So RNA polymerase binds to promoter and initiates the transcription, which is known as initiation. It uses nucleoside triphosphate as a substrate. Okay, so this is the diagram of transcription in prokaryotes. So it uses nucleoside triphosphates as substrates and polymerizes in a template dependent fashion, following the rule of complementarity. It somehow also facilitates the opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Only a short stretch of RNA remains bound to the enzyme. Once the polymerases reach the terminator region, the nascent RNA falls off. So all the RNA polymerase. So also the RNA polymerase. So this results in termination of the transcription. So an intriguing question is that how is the RNA polymerase able to catalyze all the three steps, which is initiation, elongation and termination? The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor means sigma factor and termination factor rho factor to initiate and terminate the transcription respectively. So the association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. So in bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, there is no separation of cytosol and nucleus in the bacteria. So many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. So this was all about bacteria. Now talking about eukaryotes, there are two additional complexities. There are at least 3 RNA polymerases in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. The RNA polymerase 1. So there are 3 RNA polymerase basically in eukaryotes. So RNA polymerase 1 transcribes rRNAs 28S, 18S and 5.8S, whereas the RNA polymerase 3 is responsible for the transcription of tRNA, 5S rRNA and snRNAs. snRNAs means small nuclear RNAs. The RNA polymerase 2 जो है transcribe करता है precursor of mRNA, the heterogeneous nuclear RNA. So this you have to remember, that is it. So the second complexity is that the primary transcripts contain both the exons and the introns and are not functional. Hence it is subjected to a process called splicing, where the introns are removed and exons are joined in a defined order. hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide, methylguanosine triphosphate. So methylguanosine triphosphate is added to the 5' end of hnRNA, and in tailing, adenylate residues means 200 to 300 are added at 3' end in template independent manner. So it is the fully processed hnRNA, now called as mRNA, that is transported out of the nucleus for translation. So the significance of such complexities is now beginning to be understood. The split gene arrangement represent probably an ancient feature of the genome. So the presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In ancient times, the understanding of RNA and RNA dependent processes in the living system have assumed more importance.